Okay, welcome back. This is part 2 of the bioelectricity module covering bioelectrical signals and systems. So first we're going to talk a little bit about generation of bioelectrical signals. And when we first learn about the central nervous system in our body, for example, we talk about how chemical processes are used to generate electrical signals, and these signals carry information between the brain and the rest of the body. What's interesting about this example, which is from a study completed by Tufts University in just 2011, is that for the first time they were recording patterns of bioelectrical signals which were outlining where the eyes, nose, mouth, and other features will appear. So this is still a shot of a video, but you can just kind of barely make out the eyes. There would be the nose, for example, and there's the mouth. So whereas in the previous slide we were talking about bioelectrical signals that were generated internally to the organism, in this case we're talking about the use of bioelectrical signals that are applied externally to learn something about what's going on inside the body. So in this case, this was from just February of 2013. This is a demonstration of using electrical signaling to detect the early outset of cancer in a tadpole. So whether the signals are being generated internally to the body or they're being applied externally, we need to have some way to measure them or to measure the effect of those signals. And essentially this measurement can be done using either a current, voltage, or a resistance technique. An example of looking at the measuring of resistance is with the galvanic skin response. And essentially here you're applying electrodes to the surface of the skin and measuring the conductance, which is 1 over the resistance. And this you may recognize as something that's used commonly in lie detection, for example. So the GSR will change as a function of what's going on inside the body if you're nervous, for example. Another very common measurement technique is the electrocardiogram, either the ECG or the EKG, and in this case we're making a measurement of the electrical activity of the heart. And when you're measuring these signals, as you'll see momentarily, there's frequency content that ranges as low as 0.05 hertz or 0.05 cycles per second, so very slowly varying, as high as, in some cases, 40 hertz. In the previous slide we mentioned briefly the GSR, or the galvanic skin response, and in that case the small current is passed between 2 electrodes that are applied to the body, and the resistance, or 1 over the conductance, is determined by measuring the voltage drop. In the case of the ECG, the electrodes are applied near the heart, and we're actually measuring voltages that are being generated due to chemical processes within the body. So the diagram on the left shows these several different measurement sites, V1 through V6, and if we were to look at just one of those sites, then you can see the graph on the right, which is an ECG recording of a healthy heartbeat, and on the bottom is time, and on the y-axis is the amplitude measured in steps of 0.1 millivolts. So you can see some parts of the region or of the recording that are changing very quickly with time, and some parts that are changing more slowly with time. So these would be our high frequency components, and these would be our low frequency components. And the different regions of the waveform are related to different activities that are occurring within the body. The final type of bioelectrical measurement we're going to talk about is one using a pulse sensor to measure pulse rate. And among the possible ways to measure pulse is a piezoelectric device and a photodiode measurement. So if we think back to the GSR or the galvanic skin response, in that case we were applying a current and measuring the voltage and determining the resistance. So that was a purely electrical technique. In the case of the ECG, we were measuring the voltage that's generated within the body. But in this case, with piezoelectricity and with the photodiode technique, we're having a transduction process between either mechanical and electrical, or between optical and electrical. So a piezoelectric material is one that will convert mechanical deflection to an electrical signal and vice versa. So if you think about putting some kind of piezoelectric membrane on a portion of the body, like your wrist for example, that is pulsing with your pulse rate, that will convert that mechanical deflection into an electrical signal that varies with time. We are going to focus more on the photodiode because that's the type of pulse sensor that you'll be using in your lab. And in this case, the photodiode is measuring the variation in the reflected light that's being shined onto capillaries in your finger, for example. And the amount of light that gets reflected will vary depending on the amount of blood that's in your finger, and the volume of blood is fluctuating with time as the heart beats. So here's an image of an infrared LED, for example, that would be used as the light source that would be shining on your finger, and then there's a photodiode that would be receiving the light that's reflected off the finger that varies with time and converting that into a time-varying electrical signal. And here's a device very much like the device you'll be using in the lab. This is just a clip to attach it to your ear, for example, but within this sensor there will be a light source that will shine light onto the finger, and then there's a reflected signal that gets picked up by a photodiode that's integrated on the chip, and then of course you'd end up getting some kind of time varying signal that looks like a pulse rate. And just like the ECG signal that we looked at a moment ago, there are regions of the pulse rate waveform that are high frequency and changing quickly with time, and there are regions that are lower frequency and changing more slowly with time.